Hi, everyone, and welcome back to MIMANSA Longevity's Mastermind. I'm Dr. Mima Gear, and today we're going to be talking about a really exciting topic that I think has so much interest these days, and yet we don't really know what to do with it. So I'm going to present to you all this data and some of the latest science, and the topic is personalized nutrition. So let's get started. Actually, let me do this. There we go. All right, so again, I'm Dr. Mima. This is a fall series that we're doing here on different topics related to longevity and health. This is a public mastermind that's available for the rest of the fall. It's going to be free, and then as we go into the next year, there will be a membership opportunity as well, and that'll include more protocols and specific offerings that will be unique to the MIMANSA Longevity community. So just to recap, we have 5 sessions this fall, from the fall into the end of the year into the winter. They'll be every Thursday at 7 o'clock, so you're free to join live, or you can also listen to the recording afterwards then when that gets released. This is open to the public, so if you're interested in sharing this with anybody, they can go ahead and register and get on the list, and they will receive the recordings as well. As a quick recap, last week we covered circadian rhythm and food timing. Again, a very interesting topic, so I really encourage you, if you missed that one, to go back and review it. It really is fascinating to see the circadian rhythm science and what that really represents for us as humans, and how much the circadian rhythm biology influences every single gene in our being. And this is all about light and how light essentially turns on and off genes inside your physical body. Today, we're going to be talking about personalized nutrition and the microbiome, eating for your gut and eating for your genes, and understanding your unique body type. And then the few sessions coming up, I wanted to give you a little preview. On the 30th, we'll be really diving into detox pathways. This is important for those of you who are concerned about whether or not you might have a genomic predisposition to not detox very well. This webinar and this mastermind is all about that. So make sure you don't miss it and really start to understand how your genes are influencing your detox pathways. And we'll also look at ancient purification processes that help with detoxification. The session after that will be going into November 13th, and that is a really also very much functional medicine based mastermind. Well, we'll talk about hormones and really get into the deep science around hormones and what's important to think about in terms of our hormone biology, understanding how stress and nutrition affect the hormones, and figuring out ways that we can actually interact with our hormones in a healthy, natural way to bring about more balance. And then last but not least, on the 11th of December, we'll be really focusing on the longevity blueprint. What are the key principles for longevity that you think about when it comes to this holistic longevity framework? And then taking that into next year, where we really get into protocols around longevity science and functional medicine. So I just want to remind those of you who are interested in doing a detox, we have an active detox right now taking place. It's a 21-day detox. It's not too late to join. I talked about this at the last mastermind. We'll be covering topics around gut health, hormone balance, energy and detox, sleep and recovery, weight balance, and nutrient optimization. Everyone is invited to apply to join this program. The program includes daily rhythm guides, a weekly focus theme, live support. We'll be offering 2 different Zoom classes that you can join in, and as well as a Sunday reflection. And it also includes the actual kosher cleanse framework. This is a Ayurvedic detox, and there's a framework around this, which includes Ayurveda and functional medicine, recipes, shopping lists, and a toolkit for getting your mind, body to reset. We also have a WhatsApp community that is private, that you'll be able to join and ask questions and feel supported with coaching throughout the program. For those of you who are interested for eligibility, it really is more of a wellness and longevity focus for those who want to increase their energy, not suitable for active injuries or active acute illness, surgeries, or pregnancy. There is supplement program that is included, a detox supplement program that is optional, as well as optional supplement testing. The sign-up information is available for that as well. If you sign up for this webinar, you will be receiving that in your email. All right, so let's dive into today's content. Again, I'm very excited to talk about this topic. It's been really the area of my focus in terms of my research, my training, my background. I would say since before medical school, I trained in nutrition science. And, it's been probably almost 30 years now of focus on personalized nutrition. And I'm very excited to share this latest research with everybody. And really, let's get into the conversation about what it makes sense, what is scientifically relevant. And let's also talk about some of the trends that are out there and maybe debunk some of those trends for you right now. So when we think about personalized nutrition and the microbiome, you're really eating according to your gut and your genes and your unique body type. So let's begin with the simple truth. There is no single right diet for anybody, okay? So if anybody says you have to follow this diet and it's the right diet for everyone, wrong answer. And I'll show you exactly why that's the case. The idea that there's one perfect way to eat has dominated nutrition science, nutrition programs, and really nutrition marketing for decades. And this is completely false. The calories in, calories out approach is wrong. We cannot just survive by eating less sugar, avoiding fat, and following the latest fad, right? So what we're learning today is essentially the fundamentals of new biology that is showing that metabolism is not a simple in and out math problem like a lot of people say it is. It really is a conversation. It's a conversation with your body. With your body, your mind, and your spirit. I'll even extend to say that because it really is this back and forth communication. I call it information for that reason. So one of the most elegant demonstrations of this actually came from a cell biology paper, which I'd like to present to you. This was in 2015. It showed that hundreds of people ate identical meals, the same bread, the same carbs, the same calories, and then they measured their blood glucose response. And what they found was incredibly astonishing. It was that two people could eat the exact same slice of bread and have a completely opposite outcome, right? So for one, it basically moved the needle, and for the other, the glucose would shoot up like a roller coaster, right? So here we are with two totally different responses to carbohydrate metabolism. Why? Because it wasn't just the food alone that matters. And this is where we have to change our perspective. It's not just the food alone. It's the person eating that food that matters as well, right? So we can't give a single prescription out for a certain diet. It really is personalized. So let's take a look at all the things in this cell paper that they looked at. So this is back in 2015, right? So now we're 10 years later. Can you imagine how much science has come out? People were measuring 800 people. This is a paper that came out, again, in Cell Biology. They showed the microbiome matters, the blood tests matter, the questionnaire data mattered, right? So anthropometrics, their body composition mattered, their food diary mattered. All these things went in to predict how their body responded to what's written up here, the glycemic response. Right? So it wasn't just one factor. All these things went in to then create a prediction on how somebody was going to respond to a diet that they were given of bread, simple bread. Now, we're not all sitting there eating little slices of bread, but this is just a very simple study that showed even if you just look at one thing, carbohydrate, glucose, and that's it, not all the other factors in our diet, that there are individual differences. So now let's take that a step further and look at what JAMA showed. There's a JAMA paper that validated that the microbiome actually was important as well. So it confirmed that dietary responses are individualized at the level of the individual, the host, and also their microbiome. So again, it's not the individual person, but it's the bacteria that is on that person and in that person that is also playing a role. So now you're just multiplying the personalization effect, right? So you can see this is getting to be broader and broader and broader. So when scientists integrated this information, like each person's microbiome, their genetic markers, their lifestyle factors, they found that they could actually predict the responses that their body was having much more accurately. Right? And that was a JAMA article that came out in 2019, which basically proved, so if anybody is still talking about calories, they should point them to this article. It basically proved that this model of using multiple factors, including the microbiome and lifestyle, outperformed calorie counting systems. Bottom line right there, 2019 in JAMA. So there are many things that go into deciding what right diet is for you. It is not one size fits all. So there are personalized metabolic signatures, right? Individual patterns of how your body interprets nutrition. Your gut microbiome plays a role, your genetics play a role, your hormones play a role, and your sleep patterns all play a role. They are informing your translators, right? So they're essentially translating how your body is storing, using energy, and responding to inflammation. And even, of course, now we know the gut-brain connection, how it regulates your mood. So again, it's really this harmony, this symphony that's taking place in the body all the time. So it's not a single factor. So here we are kind of looking at that big picture and trying to understand what are the levers, what are the things that matter when we look at the science. So here's what's so interesting is that timing matters. And I talked about this in the last Mastermind. The same meal that's eaten at 6 p.m. is not going to be metabolized the same at 9 p.m. It produces totally different effects depending on your circadian rhythm and what's called your per-2 clock gene expression. Again, this was a gene that we've discovered that shows that you're going to metabolize food and fat totally differently based on whether or not this gene is being expressed, okay? So in some individuals, a later dinner can stabilize the glucose. In others, it's going to spike your cortisol and trigger less sleep. So again, you can't say everyone should eat early, everyone should eat late. This is where having your genome can be very helpful. So knowing whether or not you have this per-2 gene expression can help you understand what your actual lifestyle plan should look like. So when we talk about personalized nutrition, we're really talking about, like I said, this dynamic relationship, this symphony that's taking place between your genome, your microbiome, and your circadian biology. It's a communication that's constantly taking place. And that's what we're really talking about in this mastermind. We're moving beyond the typical labels and we're getting into the why, the how, the body's feedback mechanism, because food is information. So let's talk about the microbiome and switch gears a little bit. You know, this science has come out and exploded in the last decade. But, you know, when it comes down to clinical practice, we don't yet know how to implement it. So a lot of times people have given up on this and said it's not really relevant. But that's completely false. It's highly relevant. We just need to understand what of this information is relevant and how to use it. And there will be a discovery phase for the next 10, 20 years interpreting some of these findings. So we have made a lot of progress already, and I'm going to share some of that information with you here. So the microbiome is essentially, like we said, it's everywhere. It's on your body, in your body, around you. But we're going to focus right now on just the gut microbiome. So the gut microbiome consists of about 10 to the 14 microorganisms. That's a huge amount of organisms. And you can just imagine that genome compared to the human genome, right? The human genome is microscopic compared to the microbiome genome that, again, is on and inside of us. So this is just the gut, and that's how many microbes are present. So the core mechanism that's taking place is that the microbiome is playing a critical role in nutrition. When you eat your food, specifically fermentable fibers like plant polyphenols, go through what's called a microbial fermentation process. Sometimes I hear from people they're having a hard time digesting vegetables. Well, this is why. So they essentially yield these things called short-chain fatty acids like butyrate, propionate, and acetate. And these metabolites, these short-chain fatty acids, then go on to bind to these G protein coupled receptors listed here on the actual epithelium and on immune cells, influencing the insulin sensitivity and inflammation in the body. So microbiota are influencing the body's insulin sensitivity and inflammation because of how you digest those fibers, right? So keep that in mind. The bacteria matter. Crossing the intestinal barrier, these microbiota actually act as what's called histone deacetylase inhibitors. They increase histone acetylation and modulate gene expression and gene transcription. Wow, right? These bacteria essentially produce metabolites that have the ability to cross through the intestine and affect your genome. That's incredible science. They serve as the fuel for colonocytes and support what's called tight junction integrity. We talk about leaky gut. I'm sure you've all heard about leaky gut. Well, these actual short-chain fatty acids have the ability to support this particular area of the body and help your cells regenerate. So this is very important for healing the gut. A clinical implication is that this production of these short-chain fatty acids that we talked about is different between different individuals, right? So when you look at one person, they're going to have the presence of, for example, Akkermansiae or Faecalibacterium. These are circadian rhythm bacteria, and essentially the host genomic variants in short-chain fatty acid receptors all influence the magnitude of this effect. So again, here's another great paper in very recently in Nature Metabolism that confirmed that propionate and butyrate, short-chain fatty acids, directly interact with chromatin in the DNA, increasing the accessibility of what are called promoter regions involved in mitochondrial biogenesis and cell repair. Okay, so emphasizing the importance of these short-chain fatty acids in the diet. This demonstrates that the microbiome functions as a regulatory interface between nutrition and gene expression. Again, super important, right? We're just starting to understand which bacteria, how do we use them, what's the right balance, what are different genetic factors playing a role, what is your ethnic background, your cultural factors that all play a role in diet and whether or not you're going to be responding well to a certain diet versus another. We're just starting to understand that. More studies need to be done. This is fascinating science. The microbiome converts dietary inputs into molecular signals. And these short-chain fatty acids act as epigenetic modulators with these downstream effects on metabolism, immune tone, and neurochemical balance, the gut-brain neurotransmitters right here coming from this interaction with the microbiota. So there needs to be a personalized modulation of the microbiota that is present, right? So we can't, again, all take the same probiotic. We just simply can't expect the same results. So we need to have targeted diets, prebiotics, circadian rhythm eating, and probably targeted probiotics as well to offer a viable pathway to influence what's happening at the genome level. So this is the future of science because we now know that this informs all disease, you name it, cancer, diabetes, heart disease, all of it is influenced by the microbiome. So in the context of longevity medicine, we're really thinking about how to optimize this microbial signaling that's taking place to reinforce cellular resilience and metabolic efficiency. Now let's move on to what is happening in terms of the mechanism of the short-chain fatty acids. So short chain fatty acids themselves are illustrated here: butyrate, acetate, propionate, butyrate, right? They're right here on the outside of the cell, and they're influencing what are called these GPR receptors. So they represent one of the most important signaling bridges between diet, microbiome activity, and host physiology. Now, butyrate is known because it serves as a primary energy source. It supports this barrier function, and it goes, again, beyond the gut. It has these mitochondrial effects, what's called SIRT1 signaling, that's responsible for stress resilience or stress resistance, and also insulin sensitivity. Short-chain fatty acids also affect T regulatory systems. So we're talking now about immune function modulated by short-chain fatty acids. Again, a dietary thing, something we can take in our diet and influence all these beautiful downstream effects. So there is a crosstalk between the epigenetic factors that we've talked about and the endocrine system. So again, another beautiful paper, the FEMS Microbiology Letters in 2024, showed that these butyrate and propionate molecules inhibit the HDAC activity, increasing the histone acetylation in epithelial immune cells. We know that this is how epigenetics is taking place and how we modify genes. So this goes back to this concept that genes aren't everything. It's not just the genes, it's also what we call epigenetic, which is what's illustrated here with the histone acetylation, the changes to the genes that are influenced by your lifestyle, your diet, meditation, and other holistic practices, nutrients, etc., that can actually affect the expression of those genes. It's not just what's coming from your parents. You can actively modify and change that with these methods. Now let's talk a little bit about circadian rhythm and fasting and how that plays a role here. So we know that these microbial short-chain fatty acids follow the circadian rhythm. So I said that a little while ago. They peak during the daytime feeding window, and they decline overnight. So again, going back to what we talked about last week, eating overnight, not a good idea. Working late hours and staying up overnight, not a good idea for your microbiome for this exact reason. The rhythm is synchronized with host clock genes. So the PER2 we talked about earlier in this mastermind, but also clock genes and BMAL1 gene, forming what's called a bidirectional loop between the microbial metabolism and the host, the individual's chronobiology. So we get this back and forth communication because of these genes and the connection to light. Fasting periods will actually amplify the short-chain fatty acid efficiency by improving receptor sensitivity and increasing microbial diversity, particularly in specific bacteria like Akkermansiae when we're focusing on longevity. So fasting, in other words, is beneficial. Again, at the right time, the right period of time, and I'll talk about soon in the right body type. So in practical terms, optimizing your short-chain fatty acids, we talked about all these beneficial effects of these, includes diversifying your fermentable fibers, so inulin, resistant starches, and arabinogalactans, having more polyphenol-rich foods that feed these short-chain fatty acid bacteria coming from plants, right? And then fasting windows of at least 14 to 16 hours to enhance the metabolic rhythm. And then circadian meal timing, making sure you align your microbial and host expression with the sun, with the light. So from a clinical standpoint, we're often using tests to understand this. Some of these are more research-based than others, but we're using stool metabolomics, we're using metagenomics sequencing, and we can target nutritional interventions to start to map and modify just simply short-chain fatty acids dynamics in real time. This is just one set of metabolites, right? Now, you can just imagine if you amplify that out to all the other things that we have yet to understand and study. So all these things are important. Now let's talk a little bit about microbiota diversity and individuality. So going into understanding this concept, we examine, for example, in this paper, 2 individuals that can consume the same diet and experience completely different outcomes, one that's thriving and the other that becomes symptomatic, only because of the microbial diversity and composition being different between them. So no 2 microbiomes, gut microbiomes, are identical. Even identical twins, they examined and did studies on this, only about 30 to 40% of twin microbiota in the gut are actually overlapping, right, in identical twins. So that there's a diversity here, or a lack of diversity that can also exist depending on what's happening in your body. And that diversity defines how nutrients, toxins, hormones are processed and how resilient your body is to stress. So this microbiome variability and function is what also determines your energy extraction, the production of metabolites like the short-chain fatty acids we talked about, also bile acids and neurotransmitter precursors, and your detox capacity, your body's ability to include degradation of xenobiotics like environmental toxins, and also buildup of estrogen. So when our body is going through stress, we actually build up more estrogen metabolites that need to be detoxified, and the microbiome plays a big role here. And then lastly, the barrier integrity. So we talked earlier about intestinal permeability and how leaky gut is one of the things we're often diagnosing in people, and that is influenced also by the specific microbiota, and that determines what amount of toxins your body ends up taking in, because that barrier integrity can be compromised. When it is compromised, or when you have a reduced diversity, you develop something that we term dysbiosis. In the Ayurvedic terms, it's called ama. In terms of Western medicine, it's called dysbiosis. Dysbiosis has been associated with insulin resistance, autoimmune activation, neuroinflammation, and accelerated biological aging. So again, in practical terms, if you have low microbial diversity, you can actually have a reduced capacity to produce butyrate and greater intestinal permeability. And then really, that's when we get into diabetes also, because you have exaggerated postprandial glucose and lipid response even to healthy foods, right? So getting back to like your gut actually informs a lot of things beyond your genome. It's the microbiota that kind of have a mind of their own sometimes and influence your glucose and your lipids. So let's take a look at a case. There's some divergent responses to the same diet. So we look back at the Cell paper and the JAMA paper, and it clearly demonstrates that 2 people could eat the same carbs. We talked about the bread and how different they are. And we look at the actual taxa, the microbiome, such as Bacteroidetes or Prevotella. These species of bacteria are linked to carbohydrate metabolism and inflammatory signaling. So we can identify specific bacteria that are saying, I want more carbohydrates, and they're getting you to essentially crave and eat more carbohydrates so that they can be fed. Just a simple example of something that was discovered, again, a decade ago. So microbial identity is very important, not just a caloric count, in predicting the outcome for nutrition. Now let's talk about some of the other theories and the other ideas that are out there when it comes to dietary paradigms. We talked so far about caloric intake. We demonstrated how important it is to look at the different individuality, the genome, the microbiome, and how we have this individual response. Now let's talk about some other ideas that are out there and how this fits. So right now we have quite a polarized environment when it comes to nutrition. And honestly, we always have had a polarized environment for nutrition. It's been like this since we started talking about nutrition, but it's time for this to start to change. If you look, for example, at the lectin-free diet concept by Dr. Gundry, when you look at the actual individuals, you'll find that they have an HLA-DQ or IL-6 promoter variants. So if you have these genes present, they're going to have lower levels of what are called lectin-degrading bacteria. So this is one example, Lactobacillus plantarum. And these individuals actually would benefit from having having this bacteria repleted, okay? And so others can have robust microbial degradation pathways and tolerate legumes and nightshades without issue, right? So again, it's not a one size fits all. If you don't have this particular phenotype and genotype, you likely will be able to metabolize this food just fine. So I really, caution people when they say they can't eat something. We have to understand why. We have to understand what's happening in your genome. We have to experiment and see which bacteria might be present and what we can actually replete, and then reintroduce some of the foods that you might have been reacting to before and see what your body does. It's a conversation. Another common diet right now is the carnivore diet, or also the zero fiber diet. Now, these diets can be very effective because they are essentially stabilizing symptoms. So you'll have people that are reacting often to fibers or reacting to foods, and they'll go to the carnivore diets because they're stabilizing their symptoms and they feel like it works compared to everything else they've done. Well, they temporarily stabilize the symptoms in individuals that have overgrowth or immune dysregulation by lowering an antigen exposure, right? So yes, in many individuals that have a certain dysbiotic presence or genetic influence, they will be reacting to certain vegetables and certain fibers. However, it's not just the meat that causes the change. It's the fact that you're removing the input of the antigen and you're reducing inflammation by doing so. But in the long term, I caution people on diets like this because prolonged restriction reduces what's called Faecalibacterium and Rosaburia species, and these guys are the ones that produce short chain fatty acids. Now, we talked earlier, the whole first half was all about the importance of short chain fatty acids in the research. So when you really look at this, in a logical manner, is this a proper diet for longevity? Likely not. So we're potentially compromising long-term mucosal health in exchange for reducing inflammation in the short term. Next, there are other diets that are looking just at fruits and vegetables and saying this is the only answer. Again, this can support detoxification in individuals that can have sluggish bile metabolism. So these are people with this particular genotype, or sluggish sulfur metabolism, so CBS variants, but they promote glycemic and oxalate issues for those with these other polymorphisms. So you really have to customize and individualize. And I realized as I started to study the genome and understand individual differences that when you have the power of your genome, you can actually understand what your SNPs are, what do they look like. You can start to decode this information for yourself and again, have a conversation with your diet, your body, your genome, and start to discover what can be actually taking place. So again, each paradigm can be therapeutically effective and correct depending on the individual's host genome and microbiome profile. It is not one size fits all. We really have to look at the individual and we have to look at long-term benefits. Is this the right diet for short-term or right diet for long-term? And remember that circadian and environmental modifiers play a big role here, right? So the microbiome composition is dynamic. Sleep disruption, meal timing, antibiotic exposure, that is critical. So many people have been exposed to series after series of antibiotics throughout their life. That is going to disrupt your microbiome. They say 7 to 10 years out sometimes. That's not saying don't take the antibiotics. That's saying we need to replete the microbiome after antibiotic exposure. And also in the elderly population, to increase lifespan and longevity, we really have to think about repleting that microbiome and keeping that structurally active as well for the gut. Chronic stress, I can't emphasize this enough, rapidly alters your microbiome ecology. You may think it's no big deal to gun for a deadline, but while you're doing that and you're not eating well and you're not sleeping well, you're affecting your microbiome. So in animal models, they showed that circadian misalignment alone shifted the Firmicutes to Bacteroidetes ratio within just 2 days, right? So very important that you have this relationship that's taking place. It doesn't, it's not like other blood tests like hemoglobin A1C where it takes 3 months to show up. We're talking about immediate changes with the microbiome, almost as immediate as a glucose change when you eat something. So this underscores why interventions have to include timing, rhythm, and environmental context in addition to food. Okay, so we need to think about how to apply this clinically. So from a longevity standpoint, the goal is not just to adopt a universal diet. It's to restore the adaptive microbial diversity. And that's really characterized by at least these 3 things right now based on the literature. A high functional redundancy, so multiple taxa, specific bacterial groups that are supporting the same pathway to create redundancy. Stable circadian oscillation and the microbial gene expression. So circadian rhythm, we talked last time that the body has its own clock genes that are going to get turned on and off. So do the microbiome. The microbiota have a circadian rhythm, and they also get turned on and off as well based on light. So keeping that stable and a regular pattern is so important. What time do you go to bed? What time do you wake up? We can't travel and change different time zones all the time and expect that there's not going to be a physiological response. Science is showing that that happens in any case. So there will be consequences on the microbiota. And then a balanced immune communication across the gut and brain axis. So that's the third component that is essentially something we want to restore when it comes to longevity. So the key insights here are that diet is not only is only the visible half of the equation, and the invisible half is what's happening with the microbiome that we sometimes need to test and understand. Our tests are even getting better. We can now do whole genome sequencing. It's still not really accessible for everybody yet in terms of microbiome, but we're going to get there, and it's going to happen within the next decade for sure. And determining how that diet is interpreted. Precision nutrition begins when we start mapping all of these things. And let's talk about the genes. So for those of you who've worked with me, you know how I work with the genome. This is a big part of my practice. I find there's a lot of power in understanding the genome, and especially when we start to bring in all these other variables and understand how they're influencing the clinical picture. So just a quick overview of the genome with the time that we have available to highlight some of the key pathways: the methylation pathway, fatty oxidation pathway, carbohydrate metabolism, glycemic control, inflammation and immune reactivity, histamine and biogenic amines. These are all current genes that are well described in the literature that can be used to develop individualized, personalized programs. And they're ones that I use all the time with my patients. And I love working with these because they give you so much insight into why symptoms are taking place and offer alternative pathways and how we can provide protocols for healing symptoms. And remember, there's this interplay, right, between the genes and the microbiome. Just to give you some examples, there's the FTO risk allele that many of you may have heard about, the FTO gene, This particular allele is associated with obesity, and it's moderated by gut diversity. So that's where Akkermansia became so famous and well known, because of this gene microbiome interaction that we discovered. Likewise, MTHFR related methylation deficits are amplified under dysbiosis. This is something that a lot of people don't know. So the MTHFR gene is actually amplified when you have a dysbiotic gut. And so stress responses, inability to detox properly, cancer risk, all of this is modulated by this MTHFR pathway, and that's amplified by dysbiosis. There is this bidirectional relationship that supports this idea of a hologenome, the idea that the host and the microbiome genomes function as this integrated biological system rather than separate entities. Of course, right? But yet we haven't been functioning that way in medicine. The science is showing it, and it's time for us to start practicing that way in our nutrition, in our medicine, in our wellness, and in our lifestyle. So circadian rhythm and epigenetic integration is so important as well. We talked about this in the first series, so I'm not going to get into this in too much detail, but I just want to make sure that we don't forget that part as well. And we want to align our nutrition with our genetic architecture. That is really key. So thinking about this overall loop, right? We've got this diet, microbiome, epigenome loop that's taking place. The diet shapes the microbiome metabolites shape the epigenetics, right? The epigenetics go back and influence the genome, right? So this is how we can actually start to build resilience and influence our genetics. And it gets disrupted through lifestyle patterns, through behavioral patterns, stress patterns. Again, those have also a genetic influence and a trauma influence. So stress, processed foods, circadian misalignment, they create metabolic noise. And restoring this rhythm and microbial diversity is what creates coherence. So we talk, I love talking about consciousness and coherence as part of the biological framework. At the molecular level, enzyme systems show quantum coherence and electron tunneling. So the mitochondria operate as resonant biooscillators. And the vagus nerve, the relaxational nerve in the body, is involved as well as your heart-brain coherence to directly influence the mitochondrial signaling. So all practices around meditation, coherence, heart rate variability, consciousness realms, these are all important to activate and deactivate some of these mechanisms and influence vagal tone and digestion. We're still studying how this works, but it's very promising. And this is why, because it actually has been shown in research, and it's fascinating science. Now, before we end today, I want to take a couple of minutes to talk about the Ayurvedic framework and how that's important here as well, and show some of the recent science, especially for those of you who are involved in the kosher program that are interested in working on Ayurveda with me. I love presenting the science behind Ayurveda because it's a 3,000-year-old system that is highly relevant in terms of the things we're talking about today. So this concept of constitutions in Ayurveda, Vata, Pitta, Kapha constitutions, air, fire, and earth, these are very important elemental frameworks of body types or body constitutions, and they are directly correlated with microbial profiles as well as physiological traits. So let's do a quick map for those of you who are in the program who want to understand this a little bit more in our one-on-one work together. Here's how it works. So Vata is air and ether, right? So those are the two elements that go into Vata. The physiological traits of a Vata type is variable appetite, light body type, they lose weight quickly, and irregular digestion. So that means sometimes they're constipated, sometimes they have diarrhea. It's never consistent, and sometimes, things come and go quite quickly in their bodies. So what's been proven and is so fascinating is that they have a lower abundance of this particular bacteria, Firmicutes, reduced short-chain fatty acid output, and increased gut permeability. So the Ayurvedic focus is often to have more warm grounded foods, oily foods, and reconstituting the mucosa. Again, this is an ancient science. They always had these treatments, but the science is now showing that we can map it back to bacteria and the genome. Pitta, which is a fire type, have a strong appetite, heat sensitivity, they get overheated, and a tendency to be inflamed. Now, these folks are dominant, not lower, but dominant in their firmicutes and compared to the Vata. And they have a reduced akkermansia during stress. So their antidote in terms of the fire is cooling anti-inflammatory phytonutrients, having more greens, turmeric, aloe, and stabilizing that cytokine expression in terms of their treatment. And last but not least is Kapha, which is the earth and water type. This is a slow metabolism, more fluid retention, and lower motivation. These folks have a lower diversity in their microbiome and a higher F to B ratio. So their optimal diet is light, rich in spices, and usually they'll need to have those early dinners we talked about, and enhancing their metabolic rate and microbial diversity because of the natural low diversity. So again, ancient science and our ability to apply modern medicine and create personalized protocols for individual types based on their microbiome. So these patterns reveal that Ayurvedic observation can actually form these functional clusters, and that's aligned with their nutrition. So the key focus areas as we wrap up today is that there is this biological communication. Every bite of food that you eat is having a rule set. I'm sorry, not a rule set, but a language, a communication that's taking place. And it's carrying data. So your microbiome is translating that data into metabolites. And your genes are interpreting those metabolites and expressing those patterns. Your circadian rhythm synchronizes those patterns into a coherent rhythm of repair, energy production, and understanding or cognition. So when we think about longevity, it's really important to remember these particular things. One, personalization outperforms prescription. Number two, rhythm is as important as content. So when you eat, how often you eat, right? That rhythm of it is as important as what you're eating. Microbial signaling is a therapeutic target. Genes provide the blueprint, but they're not the final verdict. And there's an integration of ancient and modern models that enhances our precision. So remember that nutrition is not linear. It's rhythmic, relational, and responsive. So finally, I'd like for those of you who are watching this either afterwards on a replay or are here live right now to do this integration practice. So I recommend maybe taking 7 days and recording a food, mood, digestive journal, but framing it through this additional lens, which I teach within the Kosha program as well, of circadian rhythm and genomic awareness. Your meal timing and the composition should be recorded every day on these 7 days journal, right? When you eat, what you eat, and how you combine certain macronutrients are all important. So record that for 7 days. Understand what your energy is doing. Notice after the meal, within 30 minutes, are you more alert? Are you more focused? Or are you more fatigued? That energy curve provides a lot of information. Understand your digestive patterns. Watch when you eat things. Are you bloated? Was that a comfortable meal? And are you going to the bathroom regularly? How are your emotions and your cognitive shifts? Food influences our emotions. Track your mood stability. Look for clarity versus restlessness. Those are key signs. Watch your sleep and your fasting windows. Document the intervals between your meals your bedtime, and your waking energy the next morning. So we actually have a whole framework here in the kosher program that I teach people, and we have a way to document all this information. For those of you at home, you can do a 7-day food journal and write all of these things down and start to reflect on this information for yourself and identify whether this is a supportive pattern, something that enhances your clarity and energy and comfort, or a disruptive pattern that consistently creates imbalance. Be aware that the entry point into precision and self-regulation is so important because awareness dictates all of it. And the practice turns into science when you start to experience this, right? So turn it into a real experience, transform the data and the embodied into what's called embodied vision so we can have consciousness and how we approach longevity. So finally, I'd just like to close and reflect that longevity is not about restriction. It's about resonance. It's about creating coherence and resonance in your body, your mind, your spirit, your connections around you, and your community, your whole life. When your genome, your microbiome, and your daily rhythm operate in harmony, you unlock a level of efficiency that is no supplement, it's not a protocol, it's really truly this amplified level of peace and harmony within your body and around you. And that harmony is the foundation of sustainable health, creativity, and vitality. So again, I'm Dr. Mima Gear, and thank you so much for attending today's Mastermind. I just want to remind you that our next Mastermind is going to be on October 30th, where we'll be diving more into detox pathways for vitality. We'll be focused on the gut, the liver, and the lymph systems and genetic influences on these pathways, looking at simple food-based detox strategies we can start to apply, as well as rituals like breath work, ceremonies, and herbal teas that support cleansing. And we'll end with a detox visualization and guided breathwork practice. So I encourage you to join next session live if you can, but if you can't, it will be available like this one over recording. And this session, like our others, will end up on our MIMANSA Longevity page, which is going to be on YouTube as well as on the Substack. So those of you who haven't had a chance to sign up, MIMANSA Longevity has both a YouTube as well as a Substack so that you can stay active and stay current in all the information that's being released. Thank you so much for following along today, and I will see you in 2 weeks.